Now 3 o'clock. Welcome everybody to RISE Learning Machines seminars. RISE is Sweden's research institute with a little more than 3,000 people working on a wide array of topics. My name is Olof Mogren and I am chair of these seminars. These seminars and the Center for Applied AI Research is part of the computer science department. This meeting will be recorded, and if you want to be removed from this recording, just let us know. Also, a nice effect of having these meetings recorded is that we have a large number of great talks on our YouTube channel, so please check that out. Today, I have the honor to introduce Professor Nataša Sladoje from Uppsala University. Nataša is the scientific leader of MIDA, Methods for Image Data Analysis Group, and the Director of Center for Image Analysis at Uppsala University. She obtained her PhD from the Center of Image Analysis at SLU in 2005, and her research interests are within the development of methods for robust image processing with high information preservation using machine learning and image processing tools for applications within medicine and biomedicine. The topic today is automated alignment of multimodal images to deep learn or not. With that, I will hand over the floor and the screen sharing to you, Nataša.
Thank you. I will hopefully manage to share my screen. And you will tell me if I succeeded.
Yes, looks good.
And I understand that you hear me, and I hope that the same holds for the others as well. So first, thank you very much for inviting me to this seminar series at RISE, and thank you for the introduction. So I will start a bit with giving context from about where I come from. So it is image analysis group at Uppsala University. We are sitting at the Department of Information Technology, and we are also hosting a Center for Image Analysis that you already mentioned, and that I have an honor to be a director for since recently. The center exists for several decades, shared by SLU and Uppsala University for a number of years, and now only hosted by Uppsala University. And we have quite many people working within image analysis. Nowadays, it is, of course, essentially, or almost all the time, also with support of machine and deep learning, which I will question a bit during this talk, but that is, let's see how it goes. We are doing research that aims at, first of all, advancing image analysis by developing methods in the field, but also supporting different types of applications where image analysis can be beneficial. Our focus is mostly on medical and biomedical applications, but also applications within digital humanities. We work with visualization and so on. So we are quite engaged in education in the field, both at undergraduate level. So we have a master program in image analysis and machine learning that I'm also very happy and proud to be coordinating. And we have a doctoral program in image analysis. And currently we have 12 PhD students, and that number can vary. We had I think like 6 graduations last year. So that is quite many people working in image analysis. MIDA, as mentioned, Methods for Image Data Analysis, is a subgroup within this context. I am leading that group. We are more maybe than others focused on general method development, while other colleagues are maybe more specialized towards medicine or life sciences, primarily microscopy and so on. So that is where I come from. And now I would like to talk a bit about our work related to multimodal image analysis and how to actually extract information from this multimodal data. And starting with the motivating example, so in, let's say, take cancer research, what I am showing here are images of tissue. The images are taken by two different modalities, second harmonic generation and bright field microscopy. While bright field microscopy is very good at showing morphology of the cells that people want to study, pathologists want to see when analyzing, when trying to detect cancer. Second harmonic generation will be very good at showing collagen structures present in this tissue. And then, of course, ideally, we would like to combine this information and to fuse the information that is present in order to have a much more informative setup to start from. Of course, in life sciences, these images are usually taken by machines that are independent, usually in different rooms, and therefore they need first to be aligned. That means to be bring in the same coordinate system to find correspondences, and then we can actually analyze the information that is present in them. So the thing is that it's quite challenging to align these images in general, and in life sciences in particular. Life sciences are quite specific when it comes to the type of data we are working with. It's very diverse data in terms of what is shown and seen in the images. Dimensionality is really high. These images are like whole slide images are hundreds of thousands of pixels, and that is quite overwhelming. Then the difference in scale is really big. So from some microscopies looking at nanometer scale compared to others that are at micrometer or even going larger. All this is, of course, the reason why we want to look at multimodal data, since all this information is really valuable, but then, of course, challenging to align. There are not so many data sets that are taken at the same time, so that's also quite specific. This imaging in life sciences can be quite it's expensive and complex, and then it's not that hundreds of data sets are taken. It can be really just a few. And of course, the problem of alignment is quite difficult, so that humans, not only that, I mean, it's expensive and demanding, it's also hard to really make it reliable to have ground truth annotations that say ground truth alignment in most of these situations. What I am showing here is a compendium of different imaging modalities that are observed in life sciences. It is compiled by a European network focused on correlated multimodal imaging in life sciences, COMULIS, and there more than 70 different microscopies are reported and described, which says something about the diversity of the data. So what am I talking about when I'm talking about image alignment or registration, how it is called? I believe that many are familiar with it, but just to be sure that the terminology and the problem is clear to all. Here I'm showing automated alignment of two microscopy images. So it is two different stains of cell nuclei, and then these cell nuclei in green and in pink are disaligned, and then are iteratively brought to match as well as they can. So it's an optimization process, and this is done by optimizing in a parameter space of a selected transformation, step by step, comparing at every step similarity or distance between the objects and trying to minimize this distance. So here we select to use affine transformation. So we are translating, rotating, and adjusting the scale of these images. And to the left, we are showing evaluation of a distance that is used to compare at every step these two images until convergence and then corresponding alignment. This starting alignment is quite big for this process to actually work well in general. And the problem with this iterative process, which essentially is optimization of a certain distance or similarity measure, is that using, let's say, gradient-based optimization usually ends up in local minima and very much depends on good initialization. This particular measure that is now depicted here is quite good, actually. We were proud to propose it. And as it is seen, it's quite smooth, but that doesn't really happen often, especially for these alignments that are as big as this one that I am showing here. So then there are these approaches that are called iterative approaches that I more or less now described that then require a selection of a similarity measure or a distance measure, allowed transformations, for example, affine here, optimization process. And if a user from, let's say, life sciences or actually in general is going looking for a suitable approach to address registration problem, then probable choice, most probable choice will be Elastic's toolbox that exists for a number of years now and allows for some quite it performs well in a general setup, allows for selection of several distance measures and takes care of a number of technicalities around. Another approach that can be taken and should be mentioned is that not the whole function is like moved and aligned, but only a couple of a suitable number of landmarks should be extracted and then matched. With multimodal data in life sciences, the problem is that such landmarks are not easy to detect and often are detected essentially either manually or even put in the samples before imaging. And then these landmarks should be paired, matched, and then the transformation can be estimated from that. EC-CLEM, where CLEM stands for correlated life and electron microscopy. So light and electron microscopy are really very much different in scale. So in these cases, point-based or landmark-based registration is a way to go. And there are, again, some toolbox that are available. But many things are here to be selected and designed and tuned to the data that we are actually working with. So, of course, a natural question in this learning and data-driven approaches nowadays comes, can we learn something of what's needed for registration? And of course, a lot can be learned, and there are different approaches taken over this time of, let's say, 10 last years, starting from learning representations of images so that alignment can be then applied on representations instead of the images themselves. Then learning similarity measures, similarity or distance metrics, so that comparison of different images in monomodal or multimodal settings can be more adjusted to the data, because those generally applicable solutions such as sum of square distances or cross-correlation or mutual information, they're a bit, well, they all have their pros and cons, but are but for so diverse data, they don't necessarily work so well. Then there are approaches to extract, to somehow learn the parameters or the deformation field, so that when given two images, we can get as an input, we can get as an output whatever is needed, either parameters of a parametric transformation or a deformation field that aligns these two images. And finally, now, due to, of course, all the problems with annotations and challenges with the manual alignments, the process is moving towards, the trends are moving towards unsupervised approaches to reach the same. And I'm listing here a bunch of, well, I would say relatively recently, over last 3 years, several surveys, quite extensive ones, that are talking about what is happening in the field of mostly medical image registration by using deep learning. So a lot is happening. And as commented, if we now kind of want to, if you're looking at contemporary models to approach registration of primarily medical data, then I would say that an average user in the field would go to, let's say, voxelmorph, one of the frameworks, toolboxes for addressing monomodal, but also multimodal image registration, where the idea is to avoid iterative optimization towards deformation field, which then allows nonlinear transformations and allows that different parts of the image are deformed differently to match each other. But then instead of that, to use, let's say, thousands of examples of image pairs, to use self-supervised approaches based on essentially some of these old ideas with where some of the traditionally used similarity or distance measures are inbuilt in the learning process, and then to learn transformations so that we can directly output the deformation field. The main goal with the learning based registration is essentially to speed it up, because the iterative process is, of course, quite time consuming, while then for a long while it was not really the performance of these learning-based methods was quite behind the iterative ones. Now things are, of course, I mean, the field is progressing and then the results are better and better, but they are still, I mean, there are issues that I will comment on along this talk. So in order to really understand what we can do, especially with respect to this, let's have methods that are generally applicable and can generalize over different types of multimodal data. We wanted to perform some evaluation looking at, let's say, 3 data sets, quite different ones, combining different modalities. So that would be satellite images of a city landscape of Zurich, a near infrared combined with RGB images. Then we had the cytology data, fluorescence combined with quantitative phase microscopy. Then we had have histology data, as already mentioned, bright field and second harmonic generation. And then in 3D, we have a data set with 4 different modalities in brain imaging. So MR, CT, and PET. And all these data sets are publicly available. The 3 of them, all but histology data, are actually acquired in so-called integral imaging. So we do have ground truth alignment from the imaging, which happens sometimes, but not often. And with this histology data, there was some manual or semi-automatic alignment performed. So we have access to these, let's say, benchmark data sets. We have alignments, and we wanted to try different methods and different approaches for this multimodal challenge to align them. And then going into, well, at the time that we started, quite natural thought was, I believe, to try to reduce multimodal approach of registration to a monomodal one, and then use quite rich toolbox for monomodal registration, which is then seen often to be much less challenging, and more is available there. This would mean that if I have a second harmonic generation to align with the bright field microscopy, the way that we would wanted to try was to, by well-known image-to-image translation approaches, first transform modality, let's say second harmonic generation, to an image that looks like bright field, but contains informative content of the second harmonic generation, and then use, let's say, try to register these expected to be simple pairs of more similar images, and therefore having more tools to work with, or to try the other direction, of course. And then we considered a number of methods based on GANs, which I'm sure you are all familiar with. So these different methods are proposed with different properties, being more supervised or non-supervised, requiring aligned pairs or not, differently treating information and content, so style and content. And then we wanted to evaluate a couple of them on the task that we are observing.
Nataša?
Yes?
Is it okay to interrupt you with questions?
Yes, however, I don't see much of the audience, so if you can help me with that, that would be great.
Sure, I will keep an eye on the chat and so on. And so with this GAN approach, did you evaluate sort of how well could you sort of transform from one modality to another? Yes. I realize that that's just a part of the problem, but how well can you do that?
Yes, and that is actually shown here. So it's a perfect timing for the question, and I'm happy that you ask it, since then I know that it's relevant and interesting. I'm showing what we managed for a bit of visual assessment first with this is then this city of Zurich data set, and also histology data. So the first column in each of these shows the original images, and then the coming 4 show what we managed to produce with, well, none of these image-to-image translation methods is ours. So we don't take neither credit nor blame for the performance. So what is to be kind of compared is that this set of images should then contain similar information as what is presented in these here, while they should all look like what is shown here. And for this quite kind of geometrically well, with a lot of structures, I think that with Zurich we are essentially not doing so poorly. Here it is maybe even more on histology data, it is maybe more visible how we are dealing with things. So I would say, I like to point to this example where we start with, we are comparing this original second harmonic generation with this column here, which is produced by Pix2Pix from Brightfield. And these are quite decent, but not really that we succeed everywhere. And of course, we didn't want to stop at visual assessment. So we compared the generated images by using Fréchet inception distance. So that is typically used distance for comparing success of image-to-image translation methods. So in this plot, which I don't want you to look in too many details, what is good is then to be to the left and hopefully high up, That would then show to the left will mean that we have a small distance, so a good transformation or good generated synthetic images. If the dots are also high up, then that means that our evaluated registration on these images is good. What we see is that we have dots which are kind of quite decently to the left, which means that we managed to have good translations at small distance, but quite poor performance when it comes to registration. So a conclusion that we have from here is, yes, we managed to get images that look decent, but they're not good enough for registration. The only thing that performs decently is kind of corresponds to these two dots, and they are not any of these image-to-image translation things, but those last columns, which are named comirs, and are actually representations of the images. So that's our own proposal in this story, and now I will go a bit more into it. So instead, observing how things work, we say, okay, we don't really need to go from one modality to the other or the other way around. Let's meet somewhere in between. Let's try to produce image-like dense representations, which will then be coming from each of the modalities, contain relevant information for registration, will be then as similar as possible. So that's what we are imposing. And then we will use these representations in some monomodal alignment framework, and then we'll get out the transformation that we can apply to the original data. And that is then animated to the right. So this learning framework is then based on contrastive learning. It requires aligned data, so it is supervised, but very due to contrastive learning and its power, very few images, one or two are enough to start with, then they're partitioned. And then we manage to actually produce these representations that then we align in different ways. An important observation was when generating comirs was that we need to take care about invariance with respect to rotation. Since generating these representations, they're generated by two independent units, no sharing weights. So they're just kind of, we learned representations that are then compared and the distance between them is minimized. But the rotated images will not produce consistently rotated representations, which appear to be really crucial for success of registration. So here, if we are looking at this animation, which then this is like stabilized view of rotations in a full circle, where what is then shown is what happens if we don't use softly imposed rotation invariance included in the loss function, or what happens when we use it. So ideally, since it's stabilized view, so like we are rotating back for this presentation, for this illustration here, we should see ideally no change over these rotations in 360 degrees, which is more or less achieved if we enable our rotation invariant part. Without it, things really change quite much for successful registration. What was good to observe was that it's enough to impose during training rotation invariance for just 4 different rotations, 90 degrees rotations, and then we can achieve quite successful invariance to all the rotations. That is shown in this plot here, where we are kind of correlating what happens with the correlating these different representations that are produced, and the orange line corresponds to what happens in in all 360 degrees, and the correlation really stays high, no matter that we don't need to impose it for every single position. Then we did some evaluation. And since we do have ground truth, we performed quite synthetic evaluation, but we felt that was the most fair. So we have an aligned pair to start from. Then we disalign it rigidly. That was our evaluation setup. And then we try to recover that transformation that we then know. So coming back to the original position, ideally we have a perfect overlap. In reality, we will have some mismatch of, let's say, landmarks, corners of the images. Then we measure this residual distance, so what is in the end disaligned. And then we say that we consider successful registration to be when this residual distance is small enough. And then we repeat the experiment many times, and then we count success. So without going into details of these numbers, let me just point to the most important things. So 3 different data sets, all in 2D, rigid displacements. This whole upper part of the table shows what happens with different image-to-image translation approaches. Once when we generate the representations, we align them either by iterative method using a specific, our own proposed distance measure, or SIFT, that would be landmark-based registration. This line corresponds to our representation learning or commerce and their alignment. And then we have a bunch of benchmark results, starting from what if we just align these multimodal images without any particular preparation using iterative approach, then some previously proposed non-learning based special similarities, and then mutual information standard for multimodal registration, and some other standard measures, such as normalized gradient field. And then we see that this is very non-stable, and the results are quite poor sometimes. So if we want to go high up, which is then success in, we can say this is percentage, what's the percentage of success, how many times we actually reach decent alignment in the end, then we can say, okay, some of these image-to-image translation methods work well. For example, on remote sensing data, when we use landmarks, then success is quite nice. However, we also see that when changing which modality we translate to which, the results are really different, because these 2 by 2 are essentially going from modality A to modality B or vice versa. So we can have 5% or 65% of success depending on in which direction we go. We can also see that most of these figures are quite low. And we can also see that, for example, histology data does not really allow for any registration by using landmarks because nothing really succeeded. We can see that our commerce perform quite well, definitely outperform all the image-to-image translation methods, also for this challenging histology data, and that they're better than any benchmarks. So we are quite happy with our COMIR-based multimodal registration. And now I wanted just to comment on an alternative popular approach, which I mentioned before. So that would be voxel morph, which we didn't even include in this table. And that is because it underperformed continuously or constantly for all our experiments. And by looking a bit around in the literature and trying to see if it's because of our mistreatment of voxel morph or for whatever other reason, we got confirmation that this is actually not so surprising, because voxel morph, for example, is designed for adjusting small deformations after images are affine aligned, so it will not recover large deformations ever. And our methods are quite robust when it comes to size of the transformation. And also when it is trained primarily on brain images in MRI. And as soon as the modalities are changed, or even organs are changed, voxel morph will not perform well at all. So efforts with voxel morph, but also other efforts on other registration frameworks that are learning-based, are really going towards trying to minimize this domain shift, essentially, like trying to make methods more robust when the modalities are changed, even different machines of the same modality, and definitely when organs are changed, and so on. I keep here in this presentation one of the nice confirmations for what we observed at talk. This is essentially a screenshot of the talk, where we hear a colleague researcher confirming that they tried even winning methods from learning-based methods from registration challenges on their data. And not only that they confirmed that they don't perform well, they also showed that classic elastics and another similar measures outperform quite continuously and consistently learning-based methods as soon as we move from the data they're trained on to something else. Okay, so we are happy with our learning-based method based on commerce and the representations. We wanted to try it in different contexts. So now just a quick run through that we can do also deformable registration with commerce. So we are now, we want to align images which are non-linearly deformed. So there is no linear transformation that will map one to the other, which is now quite visible, even though a bit synthetic on serial data. We want to produce commerce for these images, and then we need to align them, but now we just need to change rigid multimodal alignment setup to something else. That's again our proposal called Inspire. So Inspire is essentially monomodal. This is how it works, but it also presents what is the goal or what happens in deformable registration. This is now retinal images. This is what we want to register, 2 such images. We don't have this, what is shown in the middle are essentially segmented high blood vessels. That's only used for validation, for evaluation, how well this registration process is performed. But the registration is based on this type of images. Deformation field is now produced. And then when we proposed this framework that is called Inspire, and the key thing is again this specific distance measure that is inbuilt in it, we showed that we observed that it performs really well on this type of images. So comparing with the alternatives, again, iterative methods, and doing that based on Jacquard index, which then is again a measure of overlap of these structures and should be high. We see that from initial no overlap, we can get with elastics, we can sometimes get really high, but not too often. Alternatives perform similarly. Again, voxel morph didn't perform well, while the proposed method that we now have, quite kind of, let's call it basic iterative approach, is winning on this type of data. What was really important to see for us was comparison in terms of speed. As I mentioned, going to learning-based approaches, the main goal is to make these processes faster. And comparing what happens on this type of image data, which are then, there is the size like 4,000 to 3,000 pixels, then it is indeed true that voxel morph runs really fast, while classic approaches such as iterative approaches, elastic, and this unseen take quite much more time. But then our iterative method was also quite fast compared to, well, not as fast as voxel morph, but was definitely gaining in speed compared to methods that it is more related to, and that is those iterative approaches. So now we have 2 good tools in our toolbox. and that is COMIRS as representation learning, or learned representations, and then we combine that with the deformable registration approach, which is then INSPIRE. We add a bit of this invariant constraint preservation extended by also including affine and even deformable invariance, but otherwise the framework is quite similar. And then we perform very similar evaluation. Now we have 3 data sets. This is slices of MR images, so radiology data. Now these plots are then more to the left and more higher up is better, and show how, again, in these repeated experiments and evaluated residual distance after recovering known transformation, we are now even showing how not just thresholding, like we are either good enough or not good enough. Now we are showing really how much good we are. And interesting things can be observed. So the blue lines in all 3 plots show what happens if no registration is performed. So essentially show the distribution of distances at the start. If we just take 2 monomodal images from a data set and try to register, that is the best performance, the most to the left and high up in all 3, so this grayish line. Elastics is orange, and then different versions of our approach, different versions would be with different equivariances imposed, are all these other lines. And then we can see that sometimes it is better to not touch anything because things just get worse. And that can happen with elastics, which is then crossing this blue, like orange line stays even below the blue line, but can happen also with our own methods on histodata. And we can also see that when methods are used on the data that they're all most trained on or exposed to, then, and that is simply brain images, then things work quite well for elastics, for our own data approaches, and so on. When it comes to speed, we are again evaluating a bit what happens because that's important. Our framework is quite fast, while COMIR generation is really fast, and then this iterative process is a bit slower. Can be slower or faster than elastics, depending on the size and the type of data. But I wanted to show one more thing then, and that is when we were looking at this slide in this table, this is the same as I showed before. Now I want to highlight 2 things. And that is, this is our kind of our best performing method. So this is COMIR combined with monomodal registration. But then we observed this line here, which actually reports results on mutual information. And mutual information is so standard method. So it is iterative registration using mutual information, statistical measure that is simply traditionally used for alignment of multimodal data because it works with statistical distribution properties of the intensity distributions rather than their ideal matching. And then observing this good performance, which is quite kind of respectable, we wanted to explore this a bit more. And then we know that, and we also experienced that in a number of experiments, that what happens, or registration alignment based on mutual information usually is performed as I'm showing in this pair to the right, while we would ideally want it to run as it is on the pair to the left. What happens in these pairs of images, which are MRI T1, T2, so quite similar images with well-defined structures, so should be easy, is that optimization is a problem when the starting deformation or displacement is high, and our optimization simply gets stuck in a local optimum. And that is what prevents further alignment that actually reaches good performance and low difference. So we were essentially, our PhD student at that time was a bit intrigued by that. And then he decided to try to thinking that if we don't want to get into to get stuck in a local minimum or actually maximum, because we are maximizing mutual information, then it would be ideal if we could have a global optimization. So something like imitating cross-correlation maps. If we can compute mutual information for a number of different displacements of one image over the other. But unfortunately, this, because it relies on a pre-computation of several histograms and discounting, becomes really prohibitively slow. That's not feasible. So his idea was to simply do the same thing, but in a different way. So instead of computing mutual information for one position of the images, moving on and recomputing all the histograms, he was filling in the whole map of cross mutual information by actually filling in a certain bin in each histogram in all positions. So just changing the order of operations, which allowed him to express the operations as cross correlations of level sets of the intensity functions, to move to Fourier domain, to use multiplication, and to speed up things quite significantly. That is possible for translations. And then when a good translation is found globally, then it is still possible to tune a bit rotations without slowing things too much, because we are already quite close, so that rigid alignments become globally optimized and become really good. So when looking at the result of that improvement, I'm again giving the best what we could get, no matter with which image-to-image translation method. Our comirs locally, traditionally computed mutual information, and finally this what happens with the new computation. And we see that this line really looks impressive. And even more importantly, or even why that is possible at all, is because things become fast and allow for global optimization. I mean, it's the same mutual information as it was before. So speed ups due to this pre-computation are up to 10,000 times, depending on the size of the image. So let's say if you look at one alignment, once trying to align these histology images of 800 squared approximately size, when we do not look at all possible intensity levels, but we quantize them a bit, and then refine rotations in with a certain number of allowed angles, to do that directly, and that means to really do global computation of all possible displacements, would take 19 hours, while with this approach, it takes 33 seconds. And that is what allows then that this global optimization comes into place, and then that we are not really getting stuck in local optimum for mutual information, but we give it a chance to shine, and therefore we give this, we reach these really, really good results. And I don't want to spend much time on this last thing, essentially, which is just that the similar idea of let's recompute things. Let's see how we can do things more efficient, compute things more efficiently, while computing essentially what people are using. How can we go further and do things better? He tried to speed up similarly another measure that is used in medical image registration. It is called normalized gradient fields. it is suitable to align images that have quite distinct structures, so that gradients can essentially show, help to find alignment. And then illustration is shown here. So gradient fields are computed, but quite disaligned for this pair of images. And then alignment is identified nicely for, let's say, if these two images are normalized gradient field for them, and the similarity computed as simply computing cross product of these vectors, can nicely show identified good position for alignment. And then, to not go now too extensively into details, again, by expressing things in a right way, so that cross products are computed as cross correlation, which then can be moving to Fourier domain, be computed as multiplication. Things become fast, can be globally optimized. And then we go to, of course, our evaluation setup, look at our 2D images. Okay, again, image-to-image translation, COMIR, locally optimized mutual information, this previous globally optimized mutual information, and now normalized gradient field, traditionally optimized, and now new proposal. Okay, not bad, much better than it was, but not so good. Here we see that mutual information is still quite much better. And then we go to 3D and finally get to those 4 modalities of brain images. And then we register everything to everything. So we have different combinations. Now I am only showing what happens with locally optimized mutual information and normalized gradient fields. And then our own proposals. Okay, these two are, our own proposals, where this cross similarity of normalized gradient fields is the latest one, the last one I'm talking about. And we see that local optimization really fails miserably. This is like percentage of success, the higher, the better. While the normalized gradient fields optimized globally reach really fantastic results. And again, global optimization allows for not getting stuck in local optima and is essentially possible because of speeding up the process. So this is again times, and again times depending on the size of the images, which show how much faster we can go here. Also now with normalized gradient fields, much faster than mutual information in this similar setup with global computation and global optimization. So now time to come to some conclusions. And these are essentially that, yes, registration is really, really important for medical and biomedical domain. Especially in life sciences, we are exposed to so diverse data that this imposes a huge challenge for reaching a generally applicable registration approach. And on the other hand, that is really what we would like to reach. If we work with users who are not so skilled in defining and developing their own registration method for their own 2 modalities that they have, then it's very, very difficult to all the time go to a specific setup or develop something new. They would like to have something that works in more than one application scenario. What we are happy to see, being a bit traditional, and I hope that I'm not too much breaking the concept of this learning machines seminar series, I am actually highlighting that classic methods can perform really well. And it's not that we developed new classic methods, even though some of these distance measures are our own kind of development that made some things better and faster. But even algorithmic improvements, just how to compute things, how to speed things up, show to be very beneficial. This in any case does not mean that I underestimate the power of learning approaches to registration as anything else that we are, or any of the tasks we are addressing. And a lot is happening in the field, and a lot of new directions are explored, and things are, of course, improving. I still believe that this generally applicable solution is far, but we will get there, I hope. And of course, we will get there a bit easier if we get good data sets with annotations, at least for evaluation, even though, of course, supervised methods tend to perform better in general, but have their challenges, and therefore unsupervised approaches are what we are aiming for. But to be able to evaluate, to be able to understand what we are aiming for, we need annotated data. And a lot of that is recognized, all these needs and problems are recognized in the community, where there are some challenges organized annually. Learn to Reg challenge is organized since 2020 and is really trying to help that we get a bit more systematic in the development in this field, to try to somehow drive these methods to deal with learning from small data sets, to estimate large deformations, to work with multimodal data, which are challenges that are recognized, and we were trying to face them as well. Now, I don't manage to see the description and details about the winning methods of 2023, but what I saw was that at least in some aspects, the winning method is again iterative and not learning-based. So I'm really looking forward to see what others and like, I mean, there are evaluations in a number of different, with respect to different criteria. So the overall winner is, I believe, learning-based, but that's still interesting to see that the classic methods are not yet giving up. Then there is another challenge that is more in a microscopy, where the issue usually comes with huge data to align, and usually that is to align consecutive sections of tissue. So being very thin, that means deformed just by manipulation, and also not allowing for identical alignment because they're just consecutive, and being huge, that is another set of problems that are addressed by the community, but the community is moving forward. So we are expecting all the time new things and good things. And with this, I would like to thank you for listening, to conclude this story, and to give credits to the team that was working on all these methods that I have described. And to mention that all, or almost all, that I have been talking about here, the approaches that we have developed, are available in our MEDA group GitHub. So if anyone is interested, either find us or find me, and then I can help further. Thank you very much.
Great, thank you, Nataša. Do we have, or please just bring out all your questions in the audience. I can possibly start off the discussion with a question. Would you say that the sort of the solution to this will that be a hybrid solution, sort of the learning based and the iterative? Will that be the solution in the end, or do you think that sort of machine learning will get there?
Now our COMIR, or the method that I was talking about, is hybrid, since we produce representations, learn representations, and then we align them in a classic iterative way. I do believe, I mean, it was really fantastic to see this, what I showed last, at least what Johan produced with completely classic approaches. We would like to evaluate these approaches more, but they are rigid alignment so far, so 2D, 3D, but still rigid. I do believe that we will all turn to learning-based approaches and hopefully end-to-end. But it needs time, and we are suffering. People working with registration are suffering with same problem from same issues such as too much on domain shift issues and generalization issues.
Yeah. Right. Misha is asking in the chat, what is the second harmonic?
Honestly, I don't know. It's that's some particular signal that is imaged in order to visualize something that is relevant. So that's what the imaging technique is based on, and it is called second harmonic generation. So I didn't read much about what's the physics behind it.
Right.
Thank you.
Do we have one last question from Nataša? Otherwise, let's conclude there. Thank you very much for this exciting talk. Next week, we have Jonas Hellgren from RISE. He will talk about reinforcement learning, theory, toy problems, and application examples. So that will be November 16th. Welcome back.
Thank you very much.
Thank you.